is Handsome Nerd ASMR, back with another video. In this video, we're going to be going over the Yersinia bacteria using the image provided by Sketchy, and the image is called Yersin and his pets. And here is the image that we will be analyzing. So, the Yersinia bacteria, it is a gram-negative rod, and it is an Enterobacteriaceae. So, to remember that it's gram-negative rods, think about the red hues that we kind of see over here, and also in this microscopical sample of the bacteria. So the two main species you have to know include the Yersinia pestis and the Yersinia enterocolitica. But we'll talk about the Yersinia enterocolitica first. So for Yersinia enterocolitica, we're going to be focusing on the English terrier in the image that we see over here. And so this part of the image is what we'll be focusing on. And so, some distinguishing feature for the Yersinia enterocolitica is that the bacteria is motile, or very movable at 25 degrees Celsius temperature, but it becomes non-motile at the 37 degrees Celsius. So it actually likes to grow in cold temperatures. To help you remember this, think about the icicles that are outside seen in the windows that we see over here, for example. And for its main reservoir, the bacteria is zoonotic, so it is often transmitted through the puppy feces or dog poops, particularly in the outdoors. And so, to remember this, think about the English terrier that happened to poop a lot, as shown over here. And then the kids happen to be most affected by this. So, to help you remember this, think about the toddler that is next to the puppy, as shown houses, you want to utilize stool culture at 25 degrees Celsius temperature because this is the optimum growth temperature for Yersinia enterocolytica. And also do cold enrichment procedure. You can also do bipolar staining that resembles a safety pin structure. So to remember this, think about the safety pin on the toddler's diaper that we see over here. And then for treatment, most important method is supportive care. And then for anyone who is immunocompromised, such as with a low CD4 plus T cell count, you want to utilize fluoroquinolones or third generation cephalosporins for treatment. Then we have the Yersinia pestis, and so for this one we'll focus on the rodent over here. And you'll understand why after we go through these principles of Yersinia pestis. So the distinguishing feature of Yersinia pestis is that it involves bipolar staining. It is a facultative intracellular parasite, and it is actually coagulase positive, but unlike the Salmonella or Shigella, it is non-motile, or I should say, it is non-motile, and unlike the Salmonella or Shigella or something like that, it is H2S negative. So really all you have to remember is that it is coagulase positive, it is non-motile, and it is also H2S negative. And so the main reservoir for the Yersinia pestis is that it is zoonotic. And then it can be found in the southwest parts of the U.S. desert, or the U.S. deserts in the southwest part of the United States. And so it's known to be found in rodents, especially prairie dogs, chipmunks, and squirrels. And it can also be a potential biowarfare agent. To help you remember the animals where this bacteria is found, think about the prairie dog in the rodent cage that we see over here. And then for transmission, it usually occurs through the flea that is found in the wild rodents that ends up biting the human. To remember this, think about the Yersin's anti-flea spray sold here sign on the door that we see over here. Here's the anti-flea spray sign. And then this flea bite will cause sylvatic plague. And then of course there can be human to human transmission by respiratory droplets. And this is very contagious and dangerous. So for pathogenesis, it involves the coagulase contaminated mouth part of the flea. And this will involve endotoxins and exotoxins. There's also envelope antigen that inhibits the phagocytosis, and this antigen is known as the F1 antigen. This will inhibit phagocytosis. And there's a type 3 secretion system involved that suppresses the cytokine production and ultimately resists phagocytic killing. To remember the type 3 secretion system, think about the turkey pasture symbol that is used to fill up the water. So, right over here, and then let me make sure I've covered everything on this side. Okay. Yep. And so for disease, the most notable one is bubonic plague, also known as Black Death, that is due to flea bites from infected animals and later uninfected humans. So it presents with symptoms of rapidly increasing fever, conjunctivitis, and regional buboes, or very swollen lymph nodes. If you Alright. So all